today we have an exciting lecture and an exciting presenter. Adam Durbin is a member of the faculty at St. Jude Children's Research Hospital in the Division of Molecular Oncology, Department of Oncology in the Developmental Biology and Solid Tumor Program. His team works to understand how transcriptional regulatory circuitries establish malignant cell identity and how these circuitries can be perturbed for clinical benefit using conventional and novel small molecules. Today's lecture is targeting transcriptional addiction in high-risk pediatric cancers, and we're going to learn some interesting things. But with that, I'm going to turn it over to Adam, and welcome, Adam.
Thank you. Thank you, guys. Let me just share my screen. Guys, is this visible? We're good.
We can see it.
All right, great. Thank you, Jay, and thanks to Liz and Brenna and Judy Oliver and all the folks at ALSF for inviting me to speak today. That's some of the work that we've been doing over the last several years, and now continue to work on in my lab here at St. Jude. And so the title is, as you said, Jay, is Targeting Transcriptional Addiction in High-Risk Pediatric Cancers. I do not have, there we go. I have no disclosures. And before I start, I just want to point out just how prevalent Alex's lemonade stand is. So this is a picture my wife and I took when we were on vacation, and I don't know if you guys can see, we're somewhere in the Caribbean, and this is actually an Alex's lemonade stand juicer that was present at the bar. So whether you are a researcher, a clinician, or a person who is not involved with this or touched in some way by pediatric cancer, I think it's fair to say that the reach of Alex's lemonade stand is everywhere. And so I am really quite proud to be invited to give this talk today. And so as Jay said, the learning objectives for the talk today, really we're going to talk about how functional genomics has allowed us to identify transcriptional circuitries that drive malignant cell identity. I'm going to describe how these circuitries that we've defined can actually be perturbed in exciting and interesting ways to force different malignant cells to acquire new cell states. And then I'm going to talk about some of our efforts in preclinical technologies that we've been working on in order to try to target transcription and force cells into these new cell states. And so I'm a pediatric solid tumor oncologist, and initially as a resident and then as a clinical fellow, I was struck by this problem. And so this is a problem, high-risk neuroblastoma. And here you can see in this Kaplan-Meier survival curve, the overall survival probability is a function of time. This is taken from a recent COG publication. And what you can see are really two things. The first is that despite our best efforts, children who are diagnosed with this tumor still have somewhere around a 50 to 60% overall survival. And the second thing that you can take from this is that treatment ends usually around 18 months to 24 months, and children are continuing to die of this disease beyond that. So this is a clinical problem that was quite striking to me and led me to think more about. And so in combination with folks at the Broad Institute and at Dana-Farber, we used a new resource called DepMap in order to try to define new therapeutic targets for this disease, to derive both more on-target therapies as well as therapies that potentially could have reduced toxicity. We've used these new targets to define transcriptional models, and I'm showing you one here that we published in 2018, and this has actually formed the basis for a lot of our work going forward. And what I'll describe today is how we've harnessed these transcriptional models to understand how they can be perturbed to control cell state using different agents. I'm showing you here an agent, cisretinoic acid, which is clinically used in this disease, and indeed also how we can use these circuitries in interesting new ways to develop new chemicals, including one called JCLOD, which I'll describe, which has in preclinical models some very exciting new effects. And so just to start this talk, really what I'm going to start talking about is the scope of this problem in pediatric oncology and how we can use pediatric cancer functional genomics. So this is a figure taken from the SEER program at the National Cancer Institute. Here what you're seeing is a distribution of pediatric cancer in children aged under 15 years. And so what you can see is that there's a relatively even split between children diagnosed with leukemias and solid tumors. So solid tumors make up a pretty significant chunk of all of the cancers that we diagnose. And once again, here's that overall survival curve of children diagnosed with high-risk neuroblastoma. This is a particularly common solid tumor that we see, and despite our best efforts, children diagnosed with this disease really continue to display insufficient cure. And so this really tells us that we need to derive new therapeutic modalities. Now, what you don't get from this curve is what the actual realities of treatment are. And so I think it's important to highlight that children who are diagnosed with neuroblastoma will receive therapy that looks something like this. And so here you can see kids will get somewhere between 5 and 6 cycles of multi-agent chemotherapy. They'll have surgery. They'll have radiation. They'll have 2 courses of high-dose chemotherapy that is myeloablative, and then they will then go on to have autologous stem cell rescue, and then follow that with somewhere between 6 and 7 months or so of immunotherapy and retinoic acid. And at the end of all of this therapy, they have about a 60% chance of survival. And so with all of this therapy comes a number of downstream consequences, including toxicity. And so many children who are diagnosed with high-risk neuroblastoma will have long-term late effects from all of this therapy, including cardiac damage, a risk of secondary cancers, infertility, problems with growth, and hearing loss, amongst many other late effects. And so the problem isn't just that our treatments are insufficient. The problem is, in addition to this, that our treatments are extremely toxic. And so what this gets at the same point. What we really need is to derive novel therapeutics. And in order to do that, we need to know what the right targets are to go after. And so this has led many to ask questions around the mutational spectrum of pediatric cancers. And so this is a figure taken from Stefan Grosvenor's paper in Nature in 2018. And this is a concept that is very well understood in the field, and that is that in general, pediatric cancer genomes have a very low mutational rate, especially when compared with adult cancers. And what this means ultimately is that oftentimes pediatric cancers don't have a smoking gun oncogene that's mutated that allows us to derive a small molecule that we can use in order to target it. And so this leads to a key question, which is how do we identify the best targets in pediatric cancers? And from these mutational spectra, we were able to identify Jingwei Zhang's group here at St. Jude and several others identifying similar effects across pediatric cancers, that in fact the mutational spectra is actually enriched in pediatric cancers for a number of different processes, including transcriptional processes, epigenetics, MYC pathways, cohesin, and RNA processing, all involved in really regulation of transcription. And so as a fellow and now in my own laboratory, I've been really very fortunate to be part of a consortium called the DepMap Consortium. All the data that I'm going to discuss is actually publicly available at www.depmap.org. So I would encourage you to, if you don't know about this resource, to access it and look up your favorite genes. And essentially, DepMap is a functional genomics and functional chemical biology approach to trying to identify those genes that are specifically important in different types of cancer. And the approach, which is sort of illustrated here, and that is you start with a group of cancer cell lines that are thought to represent different aspects of a primary tumor. Those are profiled for their baseline genetic and molecular features using technologies like whole exome sequencing and RNA sequencing, methylation profiling, copy number variation, and so on. Those same cell lines are then taken through a combination of genetic and chemical perturbation screening. That includes chemical repurposing screens, and probably the most famous or sort of well-known approach is really whole exome CRISPR-Cas9 dropout screening. And this data, this enormous amount of data, is then fused through advanced computational analysis. And the idea here is to try to derive a therapeutic hypothesis, identify some gene that results that is in some way required in a specific cancer. And so this is a schematic of a exome-wide CRISPR-Cas9 dropout screen. Here, individual cancer cell lines are infected with lentiviruses expressing guide RNAs against a target of choice, and in this case, the guide RNAs target essentially every gene in the exome, with some exceptions. These cells are pooled, they're grown for several weeks, and then DNA is extracted. And if the guide RNA targets a gene that's required for cell viability, those cells will die. Those guide RNAs will not be found at the end when you sequence them, and so it's a dropout screen. And recently, we, along with really led by Nikesh Daria and Paquita Vasquez and Kim Stegmeyer at the Broad and Dana Farber, published the first map of these genes, which are required within different solid tumor cancer cell lines. And so here, close to a little over 80 cancer cell lines, where we now have whole exome CRISPR-Cas9 data, again, entirely publicly available. And as my lab knows very well, and all of our collaborators, we have taken advantage of this data in a number of different high-risk solid tumors, including rhabdomyosarcoma, osteosarcoma, neuroblastoma, and indeed brain tumors such as medulloblastoma, to try to understand specific genes that are required in these different tumors and how they function as a way of trying to provide insight into developing new therapies for these diseases. And so what does this data actually look like? Well, this is sort of what the data looks like when you look at it on the browser here at depmap.org. There are three different situations here, and I want to just quickly run through this. Here, what you can see is a plot, for example, for this gene, OR4S2. Each of these individual lines is a different cancer cell line. And here what you can see is that when you knock out OR4S2, the effect is mean-centered here around a gene effect score of 0. And this indicates that knockout of OR4S2 really has very minimal effects on any cancer cell line. And so this gene is essentially dispensable from that perspective. On the other hand, when you knock out this gene, TOP2A or topoisomerase, what you see is a very left shift within this distribution. Here, essentially every cancer cell line dies in response to loss of this gene. This is something we call a pan-lethal. And then what the screen was initially designed to do many years ago was to try to identify genes that are selectively required in different cancers. And so in this case, you can see there is this gene ISL1. Loss of ISL1 causes death of some cancers, but in fact has really very, very little or no effect at all in the majority of cancers. And in fact, these cancer cell lines where ISL1 loss is lethal are all high-risk neuroblastoma cell lines, and we had identified this gene, which is a transcription factor, as a dominant member of a regulatory circuitry, along with Nikesh Daria and Kim Stegmeyer and a number of other folks back in 2018. And what we now know from examining this data, especially looking at pediatric cancers compared with adult cancers, is that the processes that are enriched in pediatric cancers are different than those that are enriched in adult cancers. And in particular, transcription factors are really the number one most important group of selective dependencies in pediatric cancers within this data set as compared with adult cancer cell lines. And so harnessing this data, I want to now move on and talk a little bit about this concept of transcriptional addiction in pediatric solid tumors. And really, at this point, it probably makes sense for me to introduce neuroblastoma. I've talked a lot about it, and it's a disease that's very well known to probably most folks on this call, but I will say neuroblastoma, this is the most common extracranial solid tumor of childhood. It makes up nearly 10% of all of our childhood cancer diagnoses. And despite our best efforts, it still makes up about 15% of all childhood cancer deaths. This is an MIBG scan, which is a scan that is oftentimes done at diagnosis of patient, at patient diagnosis with metastatic neuroblastoma. This is unfortunately not the exception. This is actually what most patients look like at diagnosis. So this is a very challenging to treat disease. Neuroblastoma is an embryonal malignancy. It's thought to be derived from neural crest cells that make up the peripheral sympathetic nervous system. And much like many other pediatric cancers, it's characterized by a very low mutational rate. However, neuroblastoma is commonly characterized by dysregulation of MYC family genes, which are, in fact, transcription factors. And so dating back to the 80s, Gerard Verder and a number of other folks had identified that the MYC-N transcription factor was heavily amplified in up to about a third of cases of high-risk neuroblastoma. In addition, now we know that many other cases have very high-level expression of this gene that is independent of MYC-N amplification, caused by stabilization of the protein, and indeed, as we showed with Mark Zimmerman in Tom Look's lab in 2018, that the related c-MYC transcription factor is very highly expressed by enhancer hijacking in up to about 10% of cases. And MYC proteins, which are transcription factors, have a variety of different effects on the cancer cell, and one particular activity that they have is by amplifying transcriptional output. And so in the setting of very low-level expression of MYC-N or c-MYC proteins, these transcription factors bind to E-boxes and the promoters of target genes and drive transcriptional output. But as you drive up expression of MYC or c-MYC or MYC-N, all of these different proteins, what you get is a spillover effect. These proteins saturate E-boxes in the promoter and then move over to low affinity enhancers. And this causes a massive amplification in transcriptional output, leading to a high level expression of really all expressed genes within the cell. And so way back when, we had been interested to ask a very simple question, and that is, what are the genes that are required for neuroblastoma cells to grow that are not required for other cancer subtypes? And what we identified was a distribution of different types of genes and in fact, an overrepresentation of genes that were annotated as either transcription factors or nucleic acid binding proteins. And it turns out that this, from sort of meta-analysis of DepMap data, is a common finding. And so here you can see in neuroblastoma, transcription factors are some of the most dependent genes. These genes, ISL1, HAN2, FOX2B, and MYC-N. In Ewing sarcoma, you get FLI1 and TRIM8, which are regulators of transcription. And in rhabdomyosarcoma, you see MyoD and PAX3 as some of the most enriched transcription dependencies, all of which are involved in transcription in some way. Given that we had identified these genes as transcription factors, nucleic acid binding proteins, we wondered how they were regulated. And in fact, around the time, it had been fairly well established that there were two different modes of regulation by enhancers that drive gene expression. And so here you can see in this cartoon a typical enhancer, shown here in E, marked by a little bit of acetylation. This recruits transcription factors in the RNA polymerase complex and drives low-level expression of housekeeping genes. This is sort of a classic mode of regulation for typical enhancer-regulated genes. And we can actually see this by performing ChIP-seq to this enhancer mark, this acetylation mark, here at the RAD23B locus in high-risk neuroblastoma. And so you can see here this promoter peak really right at the start of the gene, and then upstream this very small regular enhancer. In contrast, there are these elements, rare elements within the genome called superenhancers, which are really just long chain together stretches of enhancers. And these are marked by a lot more enhancer acetylation. They bind a lot more transcription factor and a lot more RNA polymerase, and drive very, very high level expression of genes. And these genes tend to be cell identity genes and oncogenes. And so here again in neuroblastoma, you see this extensive stretch of H3K27 acetylation here at the cell identity gene in high-risk neuroblastoma GATA3. And so given that we had identified all of these transcription factors, and transcription factors, of course, play a role in cell identity, we wondered if, in fact, they were regulated by superenhancers. And we went on to demonstrate by looking at a panel of neuroblastoma cell lines and comparing their H3K27 acetyl profiles that we had resolved by ChIP-seq, that, in fact, the distribution of these genes was very similar to what we had seen for dependency, and that was an enrichment of transcription factors and nucleic acid binding proteins. And when we pushed these two data sets together, what we saw was a heavy enrichment for a very small number of transcription factors, which were both required for cell growth and proliferation, but also marked by superenhancers, and they're all shown here. And in fact, subsequently, many others have demonstrated very similar findings in other diseases, including rhabdomyosarcoma, acute myelogenous leukemia, and several others. And so how do these transcription factors work? And so we hypothesized that since these transcription factors were marked by superenhancers and in fact were required for cell growth, that in fact they might be forming something called a core regulatory circuitry. And so a core regulatory circuitry is a stable arrangement of transcription factors that control cell identity. And so here in this schematic, you can see there is a superenhancer that's driving gene expression of a transcription factor, and this produces a protein product. And you might imagine a situation where that transcription factor loops back and binds to its own superenhancer to drive its own expression. And similarly, this doesn't have to be restricted to a single transcription factor. You can have multiple transcription factors that interact. And so here you can imagine you have 3 different transcription factors, each of which is producing a protein, which is in fact a transcription factor that loops back and binds to both its own superenhancer and each other's. And this is a stable arrangement by which these transcription factors can both regulate themselves and also each other. But it doesn't just end there. In fact, these transcription factors are capable of both regulating themselves as well as going out and binding to both the superenhancers and typical enhancers of effector genes within the cell. And this is a concept that really comes from the embryonic stem cell literature, where all of the ES cell reprogramming factors are in fact members of a core regulatory circuitry. And so this is a key point here. These transcription factors both regulate each other and establish a network that is autoregulatory, but also will go out and control gene expression throughout the rest of the cell by regulating downstream effector genes. And these core regulatory circuitries can actually be dysregulated in a number of different cancer cell states. And so this is from a diagram from a paper that we have now in press at Cell Reports Medicine. And so here you can imagine a balanced core regulatory circuitry. This is sort of an endogenous cell state. You have 3 different transcription factors, and they're autoregulating themselves to establish the transcriptome. And in cancer, you can imagine a situation where you can acquire a novel oncogenic transcription factor, and this then co-opts this regulatory circuitry to a new cell fate. And this you might imagine is the situation in the case of, for example, oncogenic transcription factors like EWS-fly, Ewing sarcoma, or PAX3-FOXO, and rhabdomyosarcoma. Similarly, you might imagine that loss of a tumor suppressive transcription factor could similarly dysregulate this sort of oncogenic transcriptional circuitry. And then finally, probably the most common way that these are dysregulated is by amplification of the circuitry. And this is commonly done by MYC proteins, which invade these super enhancers and drive very high level expression of these transcriptional proteins. And this is, in fact, what we see in multiple different types of cancer, including rhabdomyosarcoma, neuroblastoma, and medulloblastoma, amongst others. And it really leads to this key question: is there, in fact, a therapeutic window? And so we had identified a collection of proteins that were selectively required in neuroblastoma cells and weren't required in very many other different types of cancer. And when we took these genes and we put them into an interaction plotter called the string database, what we found was that they were highly interconnected. And that is, in prior literature reports, these proteins were known to interact with each other. And we got this dense interaction network of all of these different proteins. In green, you can see all of these proteins that are transcription factors, and in red, you can see anything that is potentially druggable. And this led us to a model which we have demonstrated previously, whereby there are these transcription factors that form this dense core regulatory circuitry, which is invaded by MYC-N. And this is, in fact, what we see in adrenergic subtype MYC-N amplified high-risk neuroblastoma. And so over the last several years, this has really been public enemy number one. We've been interested to try and figure out ways to target this core regulatory circuitry, knowing that essentially it's a house of cards. If you target any one member of this regulatory circuitry, the entire thing will collapse, and in fact, neuroblastoma will be committed to apoptotic cell death. And we have targeted this using a number of different approaches, which I'm going to describe today. And what we know is that, in fact, it's exceptionally difficult to target transcription factors directly due to unstructured domains and oftentimes a lack of binding pockets. And so this has led several different groups to hypothesize that, in fact, a good way of going about this might be to target epigenetic proteins that regulate transcription, and that is the histone readers, writers, erasers, and chromatin remodeling proteins. And there are a number of ways of doing this, including classical inhibitors, as well as the sort of more recently described PROTACs, or proteolysis-targeted chimeras, which I'll describe later in the talk. And indeed, this has led some to hypothesize that PROTACs really are the panacea for drug discovery, now being able to take out this dragon, the undruggable target, directly to the trash. But I will point out that, in fact, we do not limit ourselves to PROTACs. We're interested in peptidomimetics, inhibitors, molecular glues, peptides, and many other different agents for targeting these different proteins. And so next, I'd like to talk a little bit about some of our efforts to think about clinical compounds and how they control and rewire these transcriptional processes. And in fact, I think of these as sort of smart bombs, targeted missiles that go after specific aspects of these wiring. Since the mid-'80s, it's been pretty well known that compounds like retinoic acid are capable of driving differentiation of high-risk neuroblastoma. So this is an image taken from Carol Teal and Pat Reynolds and Mark Israel's paper in Nature in 1985. And here you can see SMS-KCNR, which is a high-risk neuroblastoma cell line. And in the presence of retinoic acid, you get production of these neurites, which is quite indicative of cellular differentiation. And in fact, this is not just a cell culture trick patients who have high-risk neuroblastoma actually receive retinoic acid, and this has made an enormous improvement in their overall survival. And so here from Kate Mathay's New England Journal paper in 1999 reporting the very first trial in which patients were treated both with retinoic acid as well as with autotransplantation, and this led to a nearly 40% increase in event-free survival of patients. So this is a clinically actionable and important compound in the management of high-risk neuroblastoma, and yet we didn't really understand how it was that retinoic acid was capable of deriving these effects. And so in collaboration, in a project really led by Mark Zimmerman and Tom Look's lab, we went on to demonstrate that what retinoic acid does is really cause epigenetic silencing of some core regulatory circuitry loci. And so Mark took retinoic acid and treated 2 different high-risk neuroblastoma cells with it and saw that, in fact, there was downregulation of McN, GATA3, and FOXP2B, 3 members of this core regulatory circuitry. And Mark could visualize this also by performing H3K27 acetyl ChIP-seq and H3K27 trimethyl ChIP-seq. And so here at the GATA3 and FOXP2B locus, when these cells are treated with retinoic acid, they lose this enhancer acetylation and they gain trimethylation at the gene promoter and indeed over some of the enhancer elements. And this indicates that what retinoic acid was doing was in part the effects of this compound in any case were to cause epigenetic silencing. But I just finished telling you that, in fact, loss of any member of the core regulatory circuitry causes cells to die. And, in fact, that's not what we were seeing here. What we were seeing was that loss of some of the members of the core regulatory circuitry was driving differentiation. And so how could we rationalize this? In fact, Mark went on to demonstrate that retinoic acid wasn't only causing downregulation of genes. In fact, some other transcription factors were being upregulated, including SOX4 and MICE1. And this was, in fact, also associated with a gain of enhancer acetylation here in DE2C and AGP cells. You can see this gain of a new enhancer acetylation at, for example, here the SOX4 locus. And so Mark went on to perform ChIP-seq and demonstrate here at loci that were not changed in expression, other core regulatory circuitry members like HAN2, that in fact what was happening was that they were losing occupancy by GATA3 and FOXP2B, but gaining new occupancy by MICE1, SOX4, and RAR alpha. And so in fact what was happening was that the core regulatory circuitry wasn't just falling apart, it was changing. There were new transcription factor members that were becoming a part of this so-called retinosympathetic core regulatory circuitry, and this we thought was associated with differentiation of high-risk neuroblastoma cells. And Mark went on to demonstrate that actually when you treat these cells with retinoic acid, you get upregulation of these transcription factors, and this is reversible. When you wash out retinoic acid, everything settles back down, including both the upregulation of the new transcription factors and downregulation of the old members. And he could see this at the protein level. And in fact, he could also see this morphologically. When cells were treated with retinoic acid, they formed these neurites, and when you washed it out, they went back to being similar to untreated cells. And so finally, we wondered, was in fact the upregulation of these genes required for this new cell state? And so Mark generated DE2C cells with stable knockout of SOX4 and then looked to see what happened when he treated these with retinoic acid. And in fact, what he saw was a failure of these cells to drive up expression of other members of the core of this new retinosympathetic core regulatory circuitry, such as MICE1 and terminal differentiation markers like fibronectin. And so here, essentially, the message that retinoic acid causes this change in core regulatory circuitry members, leading to a new cell fate. And similarly, we had been engaged in work with folks in Colorado, Haida Ford and Kristen Artinger's group, and a very talented PhD student, Jessica Shu. And Jessica had noticed that in rhabdomyosarcoma, another high-risk pediatric cancer, that these tumor cell lines were heavily enriched for dependency on a gene called 6-1. They were highly expressed here, and you can see here the gene effect is mostly negative for these different rhabdomyosarcoma cell lines. And separately, we and several others had established that rhabdomyosarcoma was driven by a core regulatory circuitry. Folks like Javed Khan and Berkeley Grider at the NIH and Beth Stewart and Mike Dyer's groups here at St. Jude had demonstrated occupancy of a core regulatory circuitry, including genes like MyoD, McN, SOX8, and myogenin. And we had actually separately implicated this gene 6-1 as a member of this core regulatory circuitry. And Jessica performed experiments in rhabdomyosarcoma cell lines to knock down 6-1, and what she saw was, again, differentiation. And so here, RD cells, this is an embryonal rhabdomyosarcoma cell line. Here you can see in the resting state, they really don't express any myosin heavy chain, but in the absence of 6-1 proteins, all of a sudden these cells start to differentiate. And perhaps not surprisingly, when Jessica xenografted these cells or used animal models, genetically engineered zebrafish in this case, she was able to demonstrate that these cell lines were incapable of forming tumors, likely because they were terminally differentiated. And so together with Jessica, we wondered, why was it, in fact, that these cells were incapable of forming tumors, and what was controlling their differentiation? And Jessica went on to demonstrate by cut-and-run qPCR along with ChIP-seq that in these cells, when you knock down 6-1, other members of this core regulatory circuitry change their occupancy. And so here in RD cells, when you knock down 6-1, you lose the presence of MyoD at a number of different stem and oncogenic loci, and instead now you gain it at the promoters of genes that are really driving cellular differentiation. And it led to this model whereby rhabdomyosarcoma is at least in part locked in an oncogenic state because of the presence of 6-1 restraining MyoD at oncogenic loci, and in the absence of 6-1, MyoD relocalizes to drive differentiation. And so what's the message? The message here is really that rewiring or reprogramming core regulatory circuitries is really capable of producing novel phenotypes. And we can do this either chemically, for example, with retinoic acid and neuroblastoma, or genetically, as we've done in multiple different situations. And this leads to this concept that acquisition of new transcription factor members or loss of old ones leads to new phenotypes. And we're very interested to understand how this can be harnessed for therapeutic benefit, knowing that this is more now than just expression of a target, but rather the activity of that transcription factor, its localization across chromatin, and in fact, who its partners are. And so what I'd like to do now is move on and talk a little bit about some of the more novel chemical biologic techniques that we've used in order to drug transcription. And indeed, in contrast with the sort of smart bomb laser-focused approach, really we have been hard at work building really very targeted, less targeted nuclear weapons that are really designed to drug transcription. And this came out of an initial study where we had thought, is it possible to target the core regulatory circuitry by targeting transcription? And so we had noted that, in fact, all of these core regulatory circuitry transcription factors were driven by super enhancers and were, in fact, themselves transcriptionally regulated. And super enhancers are bound by large amounts of bromodomain-containing proteins like BRD4, which recruit the transcriptional complex. And these can be inhibited by small molecules like JQ1. And in fact, folks like John Maris and Kip Stegmeyer had demonstrated that this compound was effective in suppressing the growth of high-risk neuroblastoma cells. Indeed, transcriptional initiation and elongation is similarly driven by the activity of transcriptional CDKs like CDK7, which phosphorylates the C-terminal tail of RNA polymerase. And folks like Ronnie George had also demonstrated that use of THC1, a small molecule CDK7, 12, and 13 inhibitor, is capable of suppressing this dysfunction, again, in high-risk neuroblastoma. And so we had wondered what actually would happen if you put the two of these compounds, each individually with some effect in high-risk neuroblastoma. What would happen if we put the two of these compounds together? And what we got was this. And so here, by spike and microarray, we had demonstrated that combinations of JQ1 and THC1 after an hour really had very few effects on the transcriptome of the cell. After 4 hours, had many more effects and really caused global transcriptional collapse after 12 hours of treatment. And when we looked at what the identity of these genes were that were being downregulated, what we found was that after an hour, the genes that were going down the most were all the genes of the core regulatory circuitry. And so first, we were losing the core regulatory circuitry, and as we went on to demonstrate next, we were losing essentially most of the genes that were regulated directly by the core regulatory circuitry before eventually we had general transcriptional collapse. And putting these two compounds together in vitro led to very potent synergy, and we were killing these neuroblastoma cell lines. And in fact, when we did this in B2C cell xenografts, in vivo in NSG mice, what we saw was that the combination of these two compounds together really led to a striking suppression of tumor growth and, in fact, a prolongation of survival. But these compounds are pretty toxic. And so we wondered, was there a way that we could do even better? Could we improve the efficacy of this treatment and also reduce the toxicity? And so we went back to this interaction plot here and noticed that really centrally located within this interaction plot was this protein P300. And P300 is a histone acetyltransferase. This is an enzyme that has a paralog, a protein called CBP, and their domain structure is shown here. And these are multi-domain containing proteins, including bromodomains and hat domains, histone acetyltransferase domains. And these proteins, in the presence of acetyl-CoA, acetylate a number of different target substrates, and this results in the establishment of the H3K27 acetyl mark, which forms a binding spot for bromodomain-containing proteins to drive high-level target gene expression. And so we wondered, was this a potential target in high-risk neuroblastoma? And so we went and looked at DepMap, and what we saw was rather striking, and that was that the majority of neuroblastoma cell lines, each of them shown here, 19 different neuroblastoma cell lines, the majority of them were dependent, as marked by this darker red color, on P300, but really very few were dependent on this paralog CBP, despite both being expressed. And so we went on to perform low throughput knockout studies and demonstrated that when you knock out P300, you lose the target mark, H3K27 acetylation. And in fact, you also lose expression of really the dominant oncogene in this disease, MYC-N. When you knock out CBP, on the other hand, you really have neither of these effects. And this translated through into cell growth. Here we noticed that when you knock out P300, the cells grew very poorly, whereas when you knock out CBP, they continue to proliferate. And over the years, many folks have developed different compounds that target different aspects of these proteins, including compounds targeting the KIX domain, the bromodomain, and the hat domain. And we took a number of these compounds and treated neuroblastoma cells with them and demonstrated that by far the most effective compound was this histone acetyltransferase domain inhibitor called A485. And so we were left with this question, well, why, if P300 and CBP are both expressed, why do they play such distinct roles in neuroblastoma? Is it that one of them is mutated? And so we turned to publicly available databases and demonstrated, in fact, while P300 and CBP they're almost never mutated in these diseases. It's exceedingly rare. And so we didn't think that was what was going on. We knew that, in fact, there weren't strong expression differences between these proteins. And so we wondered, were they binding to and regulating different targets? And in fact, we think that that's what's going on. And so we performed ChIP-seq to P300 and CBP and demonstrated that, by and large, these proteins bound at similar loci across the epigenome, and this correlated with H3K27 acetyl and open chromatin, and in fact all of the master transcription factors of the core regulatory circuitry. But when we zoomed in specifically on the core regulatory circuitry, what we saw was really striking, and that was that P300 was there, shown in red, you can see it has similar binding as all of these core regulatory circuitry members, but really CBP was not. And so P300 and CBP both have no sequence specificity to their DNA binding. And we wondered how, in fact, P300 was getting to DNA. And usually this requires the binding of these different proteins to transcription factors. And so we took the top P300 peaks and the top CBP peaks in combination with Luca Mariani and Martha Bulick's lab and demonstrated actually that these were heavily enriched under P300 peaks for a gene called, or for a consensus sequence for the transcription factor TFAP2 beta. And when we performed ChIP-seq to TFAP2 beta, it looked exactly like a core regulatory circuitry member. It was binding along with all of the other master transcription factors. And so this led us to this hypothesis that this was actually the escort for P300 to DNA. And so we went on to ask if that was the case. In fact, we suspected that TFAP2 beta would bind to P300. And in fact, in co-IP assays, we demonstrated exactly that. And that was that AP2 beta bound to P300, but in fact did not bind to CBP. And this then led to the hypothesis that if you knocked out AP2 beta, this should lead to a failure to recruit P300 to DNA and therefore loss of H3K27 acetyl. And we went on to perform a number of different experiments showing here when you knock out AP2 beta, that's exactly what you see. You see loss of H3K27 acetyl. This is a phenotype that you do not see when you knock out other core regulatory circuitry members. But what we were left with was this idea that what we really wanted to do was to target P300 and leave CBP alone. And so here in neuroblastoma, you see P300 binding to enhancers and promoters. In normal cells, you have both of these proteins, and we had hypothesized that if there was some way that we could target only P300, this would lead to death due to loss of target gene expression in neuroblastoma. And that in normal cells, perhaps we could get some compensation by CBP continuing to function in this role. And so together with Jun Chi at Dana-Farber, we developed a small molecule that is a form called a PROTAC, or proteolysis targeted chimera. In this model, you have a small molecule that on one side binds to your target of interest, for us, this protein P300, and on the other side binds to an E3 ligase complex, including the protein here, cereblon. And this leads to polyubiquitination of your target through the E2 component of this E3 ligase complex, recruitment to the proteasome and proteasomal degradation. And so here you can see the structure of the molecule that we built, our lead molecule. Here's the bait molecule, A485, and we hooked this up to a linker and a small molecule IMID that binds to the protein cereblon. And we used this compound initially in pull-down assays and demonstrated, sort of surprisingly, that this compound was capable of physically interacting with P300 proteins, but not with CBP. And this was surprising to us because, in fact, this parent molecule, A485, interacts equivalently with both P300 and CBP. And so we went on to perform proteomics to try to identify whether this was, in fact, true, and demonstrated that after 24 hours of treatment here by SILAC-labeled nuclear proteomics, that the only significantly downregulated protein was, in fact, this protein P300, and CBP was really unaffected. And we could see this by Western blotting here over time. After 24 hours, you start to see a weakening of P300 signal, and this is concurrent with activation of cell death as marked by cleaved PARP. We went on to thinking about this schema. We went on to demonstrate that if you knock out the protein cereblon, that in fact, which we did in Kelly cells, that this actually results in the generation of an inactive molecule. And so here, cereblon is absolutely required for the activity of our protein, of our PROTAC JQuad1. We went on to perform ChIP sequencing using in Kelly cells treated with this compound. And what we saw here in these individual plots, where each individual dot is a H3K27 acetyl individual peak here at 0 hours compared to the log 2-fold change at 6 and 24 hours. What you can appreciate is that at 6 hours, we really have very few changes, if any, that are significant. And at this time point, I'll point out that really P300 levels are sustained. Here, though, after 24 hours, we have global loss of H3K27 acetylation signal, and at individual loci here, for example, at the core regulatory circuitry locus, this super enhancer that drives HAN2 expression, we have really global loss of this signal. And when we quantitated this, we saw after 24 hours of treatment, really that super enhancers were more effective than typical enhancers, although certainly in general, the H3K27 acetyl signal is quite low. And so we wondered what was this doing to cells, and perhaps not surprisingly, cells without much H3K27 acetyl and poor transcriptional output really don't grow very well. And so here, high-risk neuroblastoma cells were treated with either active JQuad, the parent molecule A485, a stereoisomer of JQuad that's inactive, or DMSO. And in fact, what we saw was that JQuad was the most effective at suppressing growth. This is really driven by apoptotic cell death that's marked by a massive induction of cells of the sub-G1 phase up to 40% after 96 hours. This similarly was associated with caspase and PARP cleavage, indicating that this was in fact apoptotic cell death that was seen with loss of the protein P300 and not with catalytic inhibition. And previously we had demonstrated that CRISPR knockout of P300 but not CBP led to loss of McN, and in fact McN is a known binding partner of CBP and P300. We wondered if in fact McN was selectively interacting with P300. We performed co-IP of these two different proteins, demonstrated that P300 physically interacts with McN, and perhaps not surprisingly, when you degrade P300 here with JQuad, you lose McN expression, whereas when you catalytically inhibit at short time periods, you really see no effect at all on McN. And so this led us to a model whereby P300 is really performing two different functions in high-risk neuroblastoma. It's establishing these lineage-specifying enhancers and with its catalytic function, and it's also forming a scaffold for McN to drive enhancer invasion. And in fact, in neuroblastoma, both of these different roles matter. And so we wondered, in fact, was this actually just a McN story? Was the whole effect really driven by McN? And so in order to get at that, we established cell lines that overexpressed McN and then treated them with JQuad. And while we saw a little bit of blunting of this effect, really it didn't explain everything. And so with John Easton's group here at St. Jude, we performed a technique called SLAM-seq to measure nascent RNA production in the presence or absence of either A485 or JQuad. So we either catalytically inhibited or degraded P300 and then looked to see what was happening to nascent transcription. And we were struck by the finding that JQuad was causing a massive downregulation of gene expression. This is whole transcriptome nascent RNA sequencing that you're seeing here with an average log2-fold change of -1 for JQuad-treated samples with really very minor effects with catalytic inhibition. And the genes that were downregulated were enriched for dependency genes. And in fact, when we went back to our string database diagram, here all of these genes individually required for suppression of or for promotion of neuroblastoma growth. What was JQuad doing? JQuad was affecting the circled genes. And so really this was a nuclear weapon aimed right at the core regulatory circuitry. So what was this doing in vivo? We wondered, could we use this in vivo? And so we established Kelly cell xenografts, and we treated these tumors in NSG mice with JQuad and demonstrated, again, a blunting of tumor growth with this first generation molecule, prolongation of survival. And what I was most struck by was really how well tolerated this compound was. We really saw very few side effects associated with this, and all of that is really detailed in this cancer discovery paper, which is online now. When we took out the tumors, we saw a loss of P300 immunostain with retained CBP immunostain. This was really enriched by gene set enrichment analysis for MYC targets, and when we zoomed in on the core regulatory circuitry in vivo, what we saw was really a loss of transcription members of the core regulatory circuitry. And so with that, I'm gonna just summarize and say really the messages from today is that lineage-specific transcription factors are really these key targets in pediatric solid tumors, and that they function to form these core regulatory circuitries that establish the transcriptional landscape and become dysregulated in cancer in a multitude of different ways. And that, as we've shown using retinoic acid and genetic approaches, but indeed our new first-generation molecule of JQuad1, that targeting these circuitries is increasingly becoming possible and is able to achieve a therapeutic window. And so this is that same schema outlining sort of where we were, starting with this clinical problem of high-risk neuroblastoma and using this to derive new therapeutic targets that ultimately has resulted in really a deepening understanding of cell state manipulation and chemical targeting. And moving forward, we're interested at developing ways to better target, to combine with conventional agents, and to understand how cell state impacts upon chemoresistance. And so with that, I'm going to essentially stop and acknowledge everybody in my lab, our collaborators, notably June Chi, Brian Abraham, Mark Zimmerman, Nikesh Daria, folks such as Tom Look, Kim Stegmeyer, and Rick Young, a number of folks externally who this really has been a team effort, and it really does take a village. Including folks like Heida Ford and Kristen Artinger, who we worked on rhabdomyosarcoma with, and a number of folks internal to St. Jude. Of course, all of our funding sources. Of course, Alex's Lemonade Stand for inviting me to speak today. And I'll just finish with one quick plug and say, if you heard something here today that you liked and you're interested, please do send me an email. My email address is here. This is our lab Twitter account, and in fact, here you can see our lab website. This is my lab here with a number of different postdocs, senior scientists, and graduate students. And hopefully, just to end the talk, I'd like to say hopefully you liked what you heard here today, and we're pretty excited about it. And hopefully you didn't think it was a lemon. And so with that, I will stop.
Wow. I like the lecture, and I like ending with a joke. That was great. Amazing talk. So we're low on time, but we have at least 4 questions in already, so maybe we could jump right in.
Sure, please.
Do you want me to read them to you, or do you see them there?
Let me see if I can pull up here we go. Yes, I can see them. So it looks like the first one says, from Charalampos Lazarus, it says, "Thank you, Adam. Nice talk. I'm wondering to what extent the dependency on core regulatory circuitry members that you see in cell lines still holds in the case of preclinical models such as mice. If this is indeed the case, are there any side effects since these factors are frequently important for lineage specification and cell state?" So this is an excellent question, and we are interested in examining these same questions. We have not personally interrogated the function of these core regulatory circuitry members in genetic models in mice, although we're certainly interested in doing that. And you're absolutely right, these are important for lineage specification and cell state. In fact, a good example of this is HAN2, which is involved in generation of neural crest cells, but also in cardiac looping. So targeting HAN2 might in fact have some effects on the myocardium. And so we are thoughtful about these sort of approaches and trying to limit toxicity, but certainly I think more to come about that. It looks like the second question's from John Bushweller. 6 transcription factors bind to the phosphatase IL-1, which is essential for their transcriptional activity. There's some tool compound inhibitors for IL-1 phosphatase activity reported. Have we tried these with neuroblastoma? We have not yet, in fact, tried these in neuroblastoma. We believe that 6 transcription factors are actually, they don't seem to be dependencies in high-risk neuroblastoma, but rather in rhabdomyosarcoma. And what I will say is that we have an ongoing collaboration with Heida Ford and Kristen Artinger to ask essentially exactly those questions in rhabdomyosarcoma. So a lot of this is hot off the presses and a lot more to come. But absolutely, we think that this is a potentially tractable approach in rhabdomyosarcoma. Another question, why do you think JQuad is specific to CBP? We think that, in fact, JQuad is specific to P300, not CBP, although we are developing, along with Jude lab, some compounds that are selective for CBP. We believe that this has to do with three-dimensional structure and how the compound forms a ternary complex between P300 or CBP and bridges this to the E3 ligase complex. Unfortunately, P300 and CBP are enormous proteins, over 300 kilodaltons, and haven't been fully crystallized. And so we've started to think about using approaches like cryo-EM in order to try and solve this ternary structure. But right now, we think that this is probably a three-dimensional interaction that is promoting binding from one to the other. Gabrielle Buchel says, the in vivo data you showed are all in NSG mice. Did you also test the protactin in immune competent model because downregulation of McM could be beneficial for immune recognition? You guys are picking up on everything that we're planning to do. So this is fantastic. So in fact, we have not yet tested this in an immune competent model. One of the issues that we identified in the cancer discovery paper was in fact that this is exceptionally challenging to do because of the fact that humans developed a mutation in the cereblon gene that actually changes substrate specificity. And so JQuad works differently in human cells than it does in mouse cells. And so we have been hard at work trying to develop immune competent models with the mutation already in there so that they're in a sense humanized to ask those sorts of questions. But we agree this is a really exciting potential avenue, especially for the potential treatment of patients with high risk neuroblastoma. Wow, they keep coming. How's your day?
Yeah, there's a few. Do you have time for a few more? There's a few more.
I'm happy to stay on and talk more. So
over in the chat, there's a couple. Beth Lawler posted one and said, "Hi Adam, really great, exciting work. Do you think the selective P300 versus CBP dependency in neuroblastoma is going to be observed in other transcriptionally addicted cancers?"
That is a great question. And so one of the things that we did to try and get at this, so the answer is yes. I think that there is going to be biased dependency on either P300 or CBP in different types of cancer. One of the approaches that we took in the cancer discovery paper was to ask, when you look across all cancer cell lines in DepMap and then also performing large screens using JQuad, what we had identified was that actually almost about a third of human cancer cell lines now are genetically dependent on P300. So in the initial demonstration, when there were many fewer cell lines in DepMap, we thought P300 was actually laser specific for high-risk neuroblastoma. But DepMap is a living, breathing thing, and as more cell lines go into this screen, we're identifying different things. And so we were actually very excited to see that P300 is now a dependency in many more different types of cancer, including things like osteosarcoma, in some cases rhabdomyosarcoma. And so we're very interested to try and pull that apart and ask, what are the things that really drive P300 dependency versus CBP, or in fact, either of those? So more to come on that, but I think that the answer to that question really is likely to be yes.